The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. So my name is Richie Porter. I'm an Associate Professor of Biochemistry at the School of Biochemistry and Immunology at Trinity College Dublin. And I'm also a member of the Biochemical Society's theme panel 3, which has a focus on bioenergetics and metabolism. So this webinar is part of our dedicated early career research program of webinars. And we are delighted to provide this opportunity today for Shubhangi, Alex, and Katherine to share their work with the molecular biosciences community. Today's webinar is titled Drug Discovery and Structure-Function Relationships. And we will hear from the 3 early career researchers who will share their current work in this field. So before I hand over to the speakers, I'd like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks if you feel like doing so. If you have a question, please type it in the question box as shown in the image on the screen, stating who the question is for, and we will try to answer as many as possible at the end. Our first speaker is Shubhangi Kandwal from Trinity College Dublin. Shubhangi is a second-year PhD student in the School of Biochemistry and Immunology, Trinity College Dublin, where I am, here in Ireland. Her project focuses on the application of rational computer-aided drug design approaches to discover novel small molecule inhibitors of conserved viral non-structural proteins of hepatitis C and of severe acute respiratory syndrome, coronavirus 2. Today, Shubhangi will discuss her work on sequence and pocket conservation across SARS-CoV-2, non-structural proteins, design of future therapeutics. So without further ado, take it away, Shubhangi.
Awesome, thanks, Richie. Is the screen visible?
The screen is there, yeah.
Okay, that's perfect. Thank you again for that lovely introduction. I'd also like to thank the Biochemical Society for giving me this opportunity today to talk about my research in the Biochemistry Focus ECR webinar series. So in this talk, I'll be talking about sequence and binding pocket conservation across the SARS-CoV-2 non-structural proteins and insight into the design of future therapeutics. Just to start, I'll start off by talking about the idea behind my project. So we've seen over the course of the COVID-19 pandemic that the majority of SARS-CoV-2 therapeutic development has focused on targeting the spike protein, the viral polymerases, and the viral proteases. As the pandemic has progressed, we've seen the emergence of different variants, starting off with the Alpha variant at the very beginning of the pandemic to the most recent Omicron variant. Many studies have reported that these proteins are prone to high level of mutation and can become drug resistant. One such example is of this study that found mutations in the 3CL proteases that resulted up to 80-fold resistance to the infamous Paxlovid drug. Not only that, it also gave the virus high reproductive fitness when compared to the wild type. Thus, it becomes very necessary that we shift our interest towards other viral proteins, such as the non-structural proteins. Not only that, it's highly necessary that we also target the most conserved residue that are present within the binding pocket of these non-structural proteins. I'm going to talk about the approach that we've used in this project in a little bit, but I'd like to first mention that our group has recently been selected to enter 2 international challenges in which we were ranked among the top 4 globally. So these challenges are organized by the CASH organization to computationally design inhibitors that would then target these non-structural proteins. So as the part of challenge 2, we were asked to target the RNA binding tunnel of SARS-CoV-2 NSP13. And as a part of challenge 3, we were asked to target the MAC1 domain of SARS-CoV-2 NSP3. So what we've done is we've kind of divided our approach into 3 phases. The first phase being the bioinformatics phase, in which we've analyzed different data sets of SARS-CoV-2 NSP sequences. So we've performed multiple sequence alignments using K-align, and we were able to identify these high conserved hotspots as well as high mutated hotspots. So anything that you see here in indigo color represents that these residues are highly conserved in nature, and the white spots here represents that these are mutations that are present within these sequences. So as an output of phase 1, we did manage to identify the highly conserved residues. So then it was time for us to then move on to the phase 2 aspect. That's the cheminformatics analysis. So we wanted to visualize these highly conserved residues in a 3D protein structure. So we've used MOE to kind of visualize and see where these highly conserved residues are present. So you can see in this picture here, the highly conserved residues are colored in green, and as I move forward from green to red, red being the residues that are highly variable in nature. Talking about the virtual screening, so we've used a tiered virtual screening protocol to screen the molecules that are present within the enamine database. So as an output of the phase 2 analysis, we had our hit list that was then sent over to the cache organization. So that's the phase 3 approach. So in the phase 3, so these inhibitors are biologically tested and experimentally validated by the cache organization. So it's really interesting to see but there's a global collaboration, really. So where the in silico prediction is done here in Ireland, and we sent over our hit list to the Ukraine, where the chemical synthesis is done, and the synthesized compound is then sent over to Canada, where the biological testing is done. And if at all, fingers crossed, we were lucky enough to get these inhibitors and know that these inhibitors do bind to these binding pockets, will be asked to perform model refinement, and this entire step would then be repeated. So most recently, we've received this good news that our hit list from Challenge 2 were synthesized and sent over to Canada for the experimental validation to begin. So I just got that tweet in as it made all of us very happy. So in this webinar, I'll be focusing on the protocol that we followed. So for both the challenges, the protocols were pretty much the same, but for this webinar itself, I'll just be focusing on challenge 3. So what was challenge 3? So it was all about finding the ligands that target the macro domain of NSP3 that competes with its substrate, the ADP ribose substrate. So I've got this snapshot here of the crystal structure that shows the ADP ribose within the binding pocket of the MAC1 domain. So what is the viral macrodomain? So the viral macrodomain is believed to hijack the host immunity by chopping off this post-translation modification that occurs on the viral proteins by the host PARP enzymes for its easy identification by the immune cells. So when this modification is removed, our immune cells are unable to identify the viral proteins. It's really smart, to be very honest. Coronaviruses are really, really smart. So the MAC1 domain is known to be highly conserved, not only across the SARS-CoV-2 sequences, but surprisingly among other group 4 viruses. So the group 4 viruses have single-stranded positive sense RNA viruses. So this kind of points out to the scope of developing pan-antivirals in the near future with these inhibitors targeting those common conserved residues that are present within the MAC1 domain. So I'll take you through now with the first phase of approach. So that's the bioinformatics approach. We have analyzed different data sets of MAC1 domain, and we found that the entire MAC1 domain is highly conserved in nature with high conservation score. We also found that the key residues that are responsible in the binding of the ADP ribose were highly conserved across the beta cove. So these residues were not only conserved across SARS-CoV-2, but were also conserved across SARS-CoV-1 and MERS. So the output again of this phase 1 was these highly conserved residues that we knew were a part of MAC1 domain. We then moved forward with the phase 2 aspect of the approach, that's the cheminformatics. So there are ample amount of crystal structures that are available on the protein data bank with extra crystal structures co-crystallized with lead-like compounds, and these lead-like compounds also have IC50 values. So we know that these actives do bind to the MAC1 domain. So as a starting point, we overlaid 22 actives. So these were like highly actives with IC50 values of equal to and less than 10.3 micromolars. And we found that these 22 crystal structures were highly similar, and there was very less movement within the X-ray crystal structures. So we moved forward with these actives. This snapshot here is the overlaid crystal structure of the 22 actives, and you can see this is the ADP binding pocket, but surprisingly these actives cover half of the ADP binding pocket, so that's the adenosine site. And what's interesting to notice is that these actives also cover and extend a bit at the top there as well. So we knew that this was our binding pocket, but we wanted to explore if there are other interesting binding pockets that we wanted to target. So this then made us wonder, and we thought of using a tool called SiteFinder in MOE. So it helps us in predicting putative binding pockets by assigning dummy atoms. So these dummy atoms are basically hydrophilic and hydrophobic cavity points. Again, interestingly, you can see here it kind of floods the central ADP binding pockets with these dummy atoms, suggesting that it's really a good binding pocket. It also flooded the site where the actives were co-crystallized, so it was suggesting that this is a really good binding pocket as well. But what we thought was most interesting were the other sites that we wanted to explore and target. So I'm going to talk about it in the next slide. So what we did was we color-coded different pockets and color-coded different dummy atoms representing our 6 binding pockets. So pocket 1 is the site where the actives were co-crystallized. Pocket 2 is just an extension really of the pocket 1, but just covering half of the adenosine site. Pocket 3 is the site just adjacent to where the actives were co-crystallized. Pocket 4 is the site where it was found that it covers half of the ribose binding site, and it's just called the ribose proximal binding pocket. Talking about pocket 5 and pocket 6, it represents the site where the ADP was co-crystallized, but just with pocket 5, we saw that it had a closed tunnel conformation, whereas pocket 6 was with an open tunnel conformation. So for virtual screening, the database that we've used is the enamine database. It has molecules with 6 to 24 heavy atom counts. So what we've done next is we've created conformations of these molecules that are present within the enamine database. We've already selected our 6 binding pockets, and then we moved forward with our virtual screening workflow. So the first round of workflow was with the FastRox volume query. So what FastRox does is it takes in the volume that is covered by these dummy atoms. We converted these dummy atoms into oxygens and carbons, representing the hydrophobic and hydrophilic cavity points respectively. So as an output, these hits kind of mapped really well with the volume that was covered by these dummy atoms. So this snapshot really shows you how well these hits mapped well with the dummy atoms and the volume that was covered by these dummy atoms. So the output of the first round of virtual screening was really an input to the second round of virtual screening. So the second round was the pharmacophore search screening. So pharmacophore features basically describes the property of a ligand that helps the ligand to make good interactions with the protein residues. So in order to generate the pharmacophore features, we used the 22 actives overlaid crystal structures and generated a consensus pH4 on MOE. But for rest of the pockets, as we did not have any actives at those binding pockets, we used an in-house software called MoPBS, which was used to then generate the pharmacophore features. Again, you can see here how well this hit maps with the pharmacophore features. So now the hit list that we have fulfills not only the fast drugs volume query, but also the pharmacophore features. So then it was time for us to then filter the hit list. So we've removed the molecules that had a molecular weight of greater than 400 Daltons, a log p-value of greater than 3.5, molecules having panes and carboxylic acid moieties. So before moving to the very last round of virtual screening, we wanted to see how efficient our docking software is. So for that, we've used an in-house software called DataPipe. So we created a haystack. So as I've mentioned, we had the 22 actives, so we know that it binds to the MAC1 domain. But to create a haystack, we wanted inactives as well. So these inactives don't actually bind to the MAC1 domain. So these decoys were then generated using DudeE. And what we wanted was our docking software to identify these actives and rank them at the top with good docking scores. And lo and behold, we did manage to find these actives being ranked at the very top with a ROC AUC value of 0.88. So in a sense, it means that it was able to identify 8 out of 10 actives. So it's like having a needle within a haystack, and Fred was able to identify these actives within the haystack that we had created. So this then brings us to the last round of the virtual screening protocol. That's the Fred docking. So we found that these hits kind of made interactions with the protein residues that were conserved across beta coves as well as our SARS-CoV-2 sequences. So the last round was then to cluster our hits. So we've used pipeline pilot for clustering. So we clustered similar looking compounds into different clusters and then extracted representative compounds by looking at their docking score and then also looking at how well they made interactions with the protein residues. So this slide here represents the entire MDG protocol of how we've screened approximately 1.4 billion molecules and how we went down to the top 150 hits that were submitted for the experimental validation using the MDG tiered virtual screening protocol. So in conclusion, the bioinformatics analysis on SARS-CoV-2 sequences suggested that there is high level of conservation within the binding pockets of NSP13 and NSP3. Our tiered virtual screening workflow has predicted hits that do make interactions with these highly conserved residues. So experimental validation results of these predicted hits are still awaited for both the challenges, and hopefully, fingers crossed, we'll receive some good news soon. If any of our hits do succeed to show activity against these non-structural proteins, we will then work on model refinement and create analogs followed by relative FEP calculations. So the future direction of my project is then to focus on other non-structural proteins and to explore the highly conserved binding pockets within these non-structural proteins. So the take-home message out of this talk is that a synergistic combination of bioinformatics, cheminformatics, and in vivo studies can help us understand this virus better and can also help us in the development of small molecule inhibitors that would bind to these conserved residues. As these inhibitors would target these conserved residues, this can lead to possible development of pan-coronaviruses inhibitor in order to kind of tackle the future upcoming outbreaks caused by these viruses. So in the end, I'd like to thank my supervisor, Dr. Darren Fain, for his constant support and guidance, the MDG group, the thesis committee, and Dr. Carsten and Dr. Fiona for their help with the bioinformatics analysis. I'd also like to thank the Irish Research Council for funding my PhD studies. And lastly, thank you everyone for your attention. And I'd be happy to answer any questions in the Q&A section. Thank you.
Thank you very much, Shubhangi. That was fascinating and nice insight into how relevant biochemistry is in the world. Okay, so we'll move on to our next speaker. Our second invited speaker is Alex Slater from the University of Birmingham. Alex is a Birmingham-based research fellow interested in the structure of platelet receptors. He received a PhD in biological chemistry in 2017 at the University of Nottingham, studying protein interactions in the contact pathway. His work on platelet receptors started when he undertook a short postdoc with Natalie Poulter, studying glycoprotein 1b receptor complex by super resolution microscopy. He then undertook a postdoc with Steve Watson, looking at structural studies of platelet receptor glycoprotein 6, before recently moving onto an independent position with the British Heart Foundation. Today, Alex will present his work entitled Zooming In on Platelet Receptors, using structural biology to understand glycoprotein 6 activation and inhibition. Far ahead, Alex.
Thank you very much. Hopefully you can all see my screen okay. I'd like to thank, yep, great. I'd like to thank the Biochemical Society for choosing to pick for me to talk today. So I'm going to be talking about glycoprotein 6 and structural studies we performed on inhibitors, which have also allowed us to look into the receptor function in more detail. So just a quick disclosure, I'm going to be talking about some nanobodies today, which we have patented. So the receptor I'm going to be talking about is glycoprotein 6, or GP6 as I'll call it. So GP6 is a platelet receptor. Platelets are anucleated cells which are present in the blood, and they have major roles in hemostasis, but also the disease state thrombosis. So GP6 is, as I said, a platelet receptor, and it's the major signaling receptor for collagen on platelets. And it induces platelet signaling through association with the FcR gamma chain. And it's a very promising antithrombotic drug target because it has major roles in thrombosis, but when you block it, you don't get any increased risk in bleeding, which is really important for these antithrombotics. So platelets are in the blood vessels. When you get an injury, collagen becomes exposed to the inside of the blood vessel. This is where GP6 will first come into contact with collagen. GP6 consists of two extracellular IgG-like domains which are involved in ligand binding. It has this long stalk, single transmembrane region, intracellular tail, and it associates with the FcR gamma chain in the membrane. And upon activation, upon binding to ligands such as collagen, it induces signaling through FcR gamma, and you end up with platelet activation and essentially thrombi form. So it says current knowledge of GP6 structure here, but actually what I mean is the knowledge of GP6 when I first started this project 5 years ago was actually very limited. We only had structures for the extracellular ligand binding domains. We had the original structure of GP6 in 2002, and then a more recent one of the extracellular domains bound to a collagen peptide mimetic. This was the paper was released in 2022, but the actual structure was deposited back in 2018. And essentially what we knew from these structures is that GP6 can form dimers. It forms dimers through the D2 domain, and GP6 dimerization has been a topic of lots of disputes over the role of dimerization, but it was believed that for ligand binding to occur, you had to have a dimer of GP6. So this is where I came in, and I came into this project back in 2018, and we had just raised a series of nanobodies against GP6. So nanobodies are single domain antibody fragments derived from camelids, and they're essentially they have the same specificity and binding affinity as full-length antibodies, but they're about 1/10 the size. And we were interested in looking at if we could use these nanobodies as potential inhibitors or as probes to target GP6. And so we got 54 nanobodies straight away from 33 binding classes. So that means that essentially we had potentially 33 different binding epitopes throughout the whole 54 nanobodies. So we did some quick characterization to see what each nanobody was doing. So I've tested one nanobody from each family, and this is a GP6 signaling assay. So essentially you transfect cells with GP6 and you're able to detect signaling levels. So under basal conditions, you get very little signaling. If you incubate with collagen, signaling goes up, and then we also added in our nanobodies to see if it inhibited or increased the GP6 signaling. And we got a range. We had some nanobodies that didn't really do much. We had some nanobodies that actually increased the binding. And then we had a few nanobodies that actually inhibited. And there were 3 nanobodies that really popped out at us being really strong inhibitors of GP6 function. And that was nanobody 2, 21, and 35. We were also able to show that, I focus here on nanobody 2 and nanobody 35 for the rest of the talk, so I don't mention 21 much. But nanobody 2 and nanobody 35 was able to inhibit platelet aggregation in response to collagen. So it was, this is confirmation that it's definitely inhibiting the receptor. And we also showed that all 3 nanobodies were able to reduce thrombus formation under flow. So this shows that if we want to prevent a thrombus formation, these nanobodies are actually very good at reducing the stability and the size of the thrombus. So our next goal was to see, okay, well, these nanobodies, nanobody 2 and nanobody 35 especially, are very good inhibitors. Can we map where they're binding on the receptor? And so that's exactly what we did. We mapped both nanobody 2 and nanobody 35 using X-ray crystallography. And we showed that they bind the exact same pocket on GP6, and that pocket is just adjacent to where collagen binds. So collagen binds to this pocket here and here, and you can see the nanobodies sit right next to it. So essentially they just sit right next to the ligand binding site, and both nanobody 2 and nanobody 35 utilize this key arginine 46 residue on GP6. And I showed in a surface binding assay that if I mutate this arginine 46, we no longer get any binding at all of GP6 to the nanobodies. And you can see both nanobodies are binding to this nicely conserved pocket on GP6. So it's just by pure luck that they just happen to be binding to the exact same space, the exact same binding site. Now, the other thing we found, and we mapped the binding sites, which is why we did this, but actually both structures gave brand new conformations of the receptor itself. So not only are we looking at sort of characterizing inhibitors, but these inhibitors are also allowing us to look at new conformations of the receptor itself. So first off, nanobody 2, within nanobody 2 bound structure, we actually got a brand new dimeric structure. So these are the previous dimeric structures. You get this back-to-back dimeric structure with the D2 domain forming this kind of beta sheet interaction. However, we got a domain swap, which is essentially the D2 domain from one subunit threads itself into the D2 domain of a second subunit, and we end up with a domain swapped dimer, which was rather interesting. And then we did a quick experiment where I took this loop region here. This loop is critical for the formation of this domain swap, and I actually truncated it. And essentially, if this loop here in the original structure is not critical for the structure, so essentially if the top structure is real, this truncation wouldn't really do anything. Whereas if our domain swapped conformation is a real biologically active or biologically significant conformation, then we would see some effects with this truncation. And what I show is that the wild type protein, and again, this is an NFAT signaling assay where I can detect GP6 signaling in cells. In the presence of collagen and the collagen peptide mimetic, we get an increase in signaling. However, the truncated mutant, we get no signaling at all. So this is essentially a signal dead mutant, which potentially shows that this domain swap is a biologically active conformation of this receptor. And then secondly, the nanobody 35 structure, which is the second structure that I got, was actually a monomeric conformation. Now, as I said before, all the structures previously have been of dimeric structures, and this particular structure actually gave a monomeric GP6. And now going back to a comment I made previously where it was believed that dimerization of this receptor was required for ligand binding. Well, actually, if you overlay the monomeric structure with a single subunit from the dimeric structure, the ligand binding sites are unchanged, the exact same binding pocket. So there doesn't seem to be any conformational change that occurs when you actually dimerize the receptor. And secondly, this comes from another group, actually, the group that solved the structure bound to the collagen ligand. And they showed that actually a GP6 dimer can simultaneously bind to two collagen triple helices, and that's due to the way collagen bundles in parallel bundles. And you can see the angles here don't match up with binding to a parallel bundle. So not only is the ligand binding site not affected by being a dimer, the dimer can't even bind to ligands simultaneously. So this led us to completely reimagine what we know about GP6 dimerization, because we know it exists. GP6 definitely dimerizes. We've shown this in cells and in platelets, but we don't know why. And this led us to propose a new model whereby GP6 is present as a mixture of monomers and dimers in the cell. And then when you cluster GP6 with a multimeric ligand such as collagen, you cluster the FcR gamma chains, which are responsible for signaling, and it's that clustering which is initiating a signal. And essentially, if you cluster dimers compared to clustering monomers, you just increase that critical mass of FcR gamma chains, which essentially just means you get more signaling. So we believe that GP6 dimerization is actually important for just for the cluster size as opposed to any conformational changes. And then the final structure I want to share with you is actually one of another sort of antibody-based drug. This one's glensosumab, which is a Fab fragment, and this is an inhibitory Fab fragment that binds to GP6. And this has been developed for many years now, and it's actually just completed phase 2 clinical trials with positive results. So it's a very potentially very exciting drug that may be coming out, but it's never been mapped. And so I was involved in the mapping of this drug, glensosumab, to GP6. And unlike the nanobodies, which bind to the D1 domain, glensosumab actually binds to the D2 domain. And if I overlay glensosumab, which is in orange and blue here, if I overlay that with the GP6 CLP structure, what you see is that although the Fab is binding away from where the ligand binds, because the ligand is binding to the D1 domain, just the sheer bulk of the Fab gets in the way of the collagen chain. So it does block ligand binding indirectly, but because it's binding to D2, and we know D2 is involved in dimer formation, it actually also blocks dimerization. And we show this using fluorescent correlation spectroscopy, whereby if you take GP6, which is present as a mixture of monomers and dimers, incubate it with glensosumab, you get a reduction in molecular weight, which is closer to the monomeric control. So it clearly is preventing dimerization. And then in my title, one of the things was trying to move beyond, move towards the full-length structure of GP6, because we're looking at very small fragments. We're only looking at this portion in all our structures. So I really wanted to go ahead and try and solve the structure of the full-length complex, in particular bound to the FcR gamma chain, to get that whole complex. And this is important because it allows us to really look at how GP6 dimerizes in the membrane, not just in recombinant form, but actually in the membrane. It allows us to see how it associates with the various chains and other proteins that GP6 is associated with, such as tetraspanins. How activation occurs, is there a conformational change? We think not, but again, we're only looking at recombinant proteins. And are there any other sites that can be targeted for drugs? So the way that we're approaching this, and this is my last slide, is we are using styromalic acid. And when you incubate styromalic acid, which is a long polymer, it actually forms this uniformly shaped ring when incubated with membranes, and it essentially chops out your protein from the membrane, including the phospholipids surrounding it. So it's a very kind of native way of extracting membrane proteins from the membrane compared to other methods such as detergents. And so we incubated GP6 FcR gamma transfected cells with the styromalic acid. We were able to extract and purify out the protein, and we show using gel filtration that we get multiple different species present, from big to small. Using native page, we were able to see that actually we get a mixture of monomers, dimers, and higher order oligomers present within the cell. And then we took it to negative stain in order to see what the particle quality was. Particle quality wasn't great. I think the sample was too heterogeneous, but we do show that these particles do align along collagen fibers. So the GP6 present within these SMAPs are functional and still able to bind the ligands. So we're moving forward now to try and improve particle quality in order to hopefully start getting some structural information out of this complex through cryo-EM. So in conclusion, we've characterized and identified a series of potent inhibitory nanobodies that can be used as antithrombotics for GP6. By mapping the binding sites, not only did we, one, find out where they bind, we also found two novel conformations of the receptor. So it's linking drug discovery with structure function. We've identified another blocking agent in glycosumab that inhibits both dimerization and ligand binding, which offers another way of another mechanism of inhibiting this receptor. And now we're moving towards looking at the full-length complex of GP6 in the membrane. I'd like to thank everyone at the University of Birmingham, University of Nottingham for helping out with the crystallography, and lots of other people who helped out with this project. And also thanks for listening, and I'll be happy to answer any questions towards the end.
Superb. Thank you very much, Alex, for a fascinating lecture. And looking forward to the questions for you at the end. So our third invited speaker is Katherine Peters from AstraZeneca. Katherine is a research scientist working in the R&D section at AstraZeneca. After completing a biochemistry degree at the University of Oxford in 2021, she started a role in the Discovery Sciences protein science team based in Cambridge. In her team, they express and purify recombinant proteins to support downstream work in early stage small molecule drug development, primarily supporting projects in the oncology therapy area. And today, Katherine will present her work titled Recombinant Protein Production for Drug Discovery at AstraZeneca. Take it away, Katherine.
Thank you, and thank you for coming to listen to my talk today. So my name is Kathy, and I'm a research scientist in the protein sciences team at AstraZeneca, and we're based in Cambridge in the UK. And today I'm going to talk to you about how we produce recombinant proteins for drug discovery at AstraZeneca. So I'm going to start by giving a quick introduction as to where we sit within the company. So we're part of Discovery Sciences in biopharmaceuticals R&D, which is led by Mike Snowden. And Discovery Sciences is responsible for identifying novel targets and identifying and validating new hits against these targets in a number of therapy areas. So our therapy areas are oncology, cardiovascular, renal and metabolism, respiratory and immunology, and rare diseases. So within Discovery Sciences, we sit in Discovery Biology, which is led by Steve Rees. So in Discovery Biology, we are responsible for producing high quality assays and reagents to support the early stages of drug discovery campaigns. So what is our role within the protein sciences team? So in protein science, we have the aim to produce high quality protein reagents to support the early stages of campaigns to find small molecules against a number of protein targets. So in the UK, we primarily produce proteins to support projects in our oncology therapy area. And we have another team, protein sciences team based over in Sweden. They produce proteins for the other 3 therapy areas. So in the drug discovery pipeline, we sit at the very early stage of it. So our proteins are used for lead generation and lead optimization activities, and they're used for a wide range of downstream activities. So this includes the development of biochemical assays to test protein function and possible compounds, for various hit finding approaches, so whether that be a full high throughput screening campaign or a DNA encoded library screen, for various structural biology approaches, and for the iterative cycles of design, make, and test. So once we produce a protein, that doesn't mean that's the last we ever see of it. A really nice thing at AstraZeneca is that our project teams are interdisciplinary. So that means that a member from each of the different scientific areas sits within a project team. This means that we can see the results of an assay or a structural campaign that was run with our protein. And we can also provide input to the project team as to how we should progress forwards after achieving certain results. So I'm now going to give a quick overview as to what our general workflow looks like. So before a project can begin, we must first assess the feasibility of producing protein reagents. So we look at the literature and in-house precedent to have a look at the feasibility of protein production and what sort of scales we might be able to achieve. This allows us to give realistic timelines to the project teams and also identify and highlight potential challenges that may arise before the project starts. However, once a project starts, we must then design the constructs. So this is done using a combination of personal experience. So everybody probably has a favorite tag they like to work with, the literature precedents, but also the hit finding approach. Because depending on what the project team wants to achieve and what hit finding approach they want to use, different domains of a protein might be required, different tags might be needed, and various interaction partners could also be needed. So our constructs are then built by the molecular biology team, which is over in Sweden. And once we have received our constructs, we put them through a robust standardized small expression screening system, which allows us to screen hundreds of constructs every year and is really important so that we can optimize the conditions of expression before committing resources to large-scale expression and purification. So our expression screening involves expressing the protein on a small scale and then lysing open the cells. We then use a phenexis, which has, and we have these tips with affinity resin contained within them so that we can capture the protein, wash the tips, and then elute the protein. And then we use STS page gel to quantify the estimated yields. So this is important to us because it allows us to estimate yields we might likely be able to achieve when we move to a large scale. We can optimize the expression conditions here before committing resource. And then lastly, if we have a series of constructs, maybe with different boundaries or solubility tags, we can select the best expressing constructs to take forward for large scale purification. So for large scale expression, we use either E. coli, insect or mammalian cells to express our proteins. We then use active pure systems. So these have multi-valves which allow for multiple loops and columns to be used in parallel, allowing us to purify proteins in a semi-high throughput manner. So as a standard workflow, we would use affinity chromatography, maybe then tag removal and reverse affinity, potentially an ion exchange step, and then lastly we polish off the protein and remove any aggregates using size exclusion chromatography. So at this stage, significant scientific input is required and often a lot of optimization is needed to get high quality protein reagent. And then lastly in our workflow, before we send our protein to its end use, we want to ensure that it is of high quality so that we can believe the results that are achieved downstream. So as a minimum, this includes the concentration using a nanodrop, the chromatogram from size exclusion, so that would be the A280 trace. We look at the purity by STS page gel and the protein mass using intact protein mass spectrometry. In addition to this minimum QC setup, we also have the ability to do peptide mapping, DLS and DSF if required as additional modalities. So in our workflow, we do also face a number of challenges through working with proteins. So the first of this is not only do we have to produce proteins of high quality, we also have to produce high yields. So this can be multiple milligrams of protein for a request. Then next, our project portfolio is becoming increasingly complex. So we're faced with having to express and purify unprecedented or highly complex and challenging targets. And then because our protein resources are often a limiting factor at the start of a small molecule campaign, we often have to work to short timelines so that other downstream users in the project team can have their reagents. And because we produce proteins for so many different end uses, they all kind of have slightly different requirements. So the domain boundaries for an assay versus a structured crystallography system might be different, or you might need the protein tagged or in a different state. So we often, whilst working with the same protein, have to work with multiple constructs of this protein in parallel. And even small changes to a construct can change the way a protein behaves. And in relation to this is variety, because proteins don't exist in isolation in biology. So we may have to look at multiple isoforms of a protein if a project wants to try and achieve isoform specificity, or perhaps purify multiple components of a complex or pathway in the cell. And then lastly, it's really important to have robust structural systems in place to support structure-based drug design, and this can be especially challenging if there's no precedent for structural systems of a target. So to summarize, the protein sciences team and discovery biology at AstraZeneca produce high quality protein reagents for the earliest stages of the drug discovery pipeline, and our reagents are used to support a number of downstream applications. Before we purify proteins, our constructs go through standardized expression screening systems so that we can optimize the expression conditions before committing resources to purification. And we use standard purification techniques to purify our proteins. And then our workflow faces a number of challenges, particularly as we have to produce high quality proteins in high yields to short timelines. And we are faced with an increasingly complex project portfolio. So I'd like to thank the Biochemical Society for giving me the opportunity to present on our workflows today, and also thank the Protein Science and Dow Screening team in Cambridge. They're a really great team to work with, and I'm happy to answer any questions in the upcoming Q&A.
Oh, thank you very much, Kathy. That was a great insight into a fascinating world. All right, so we can now welcome questions for any of our speakers. If you have a question, please type it into the question box on the right-hand side there, shown on the image on the screen. I don't see any questions there at the moment. So maybe while people are thinking of questions, I'll ask a few questions that I have written down here. So if I go backwards from Kathy first, so I note that you express in E. coli. So, I mean, I've expressed many protein in E. coli, and I would encounter problems like proteins ending up in inclusion bodies and having to extract them and getting the right conformation. Is that an issue for you, or the protein's just so small that it's not a problem, or what?
So E. coli we use as standard, and then if the protein, so a lot of our targets do work in E. coli, or we could maybe optimize the expression conditions, or we have, because we have the option to move to insect cells, if E. coli are proving problematic, we can move over to insect cells to express protein.
Okay. And then the second question, which sort of links to that, would be, I suppose it links to what Alex was talking about, is glycosylation. I mean, glycosylation obviously would occur at possibly relevant sites if you're expressing it in a mammalian cell, like a Chinese hamster ovary cell or something. So how do you just circumvent that problem, or is there glycosylation by insect cells?
To be honest, it's not something a problem I've had to deal with in my time yet, so I'm not entirely sure what the answer would be for that.
Yeah. Okay, that's grand. Thank you, Katherine. Alex, then I suppose while I'm waiting for questions to come in, I'll ask Alex a question or two. So the camelid nanoparticles, these heavy chain antibodies, is there any suggestion that our body might actually trigger an immune response against those nanoparticles?
So it's a good question, but they're actually very non-immunogenic, because the actual nanobody is, I'd say, 95% similar to all the antibodies we have. So the only difference between one nanobody and any other variable chain antibody is just the 3 CDR binding loops. So they're so similar that they don't cause any major immunogenic response.
Okay. Let me just see if there's any other questions at the moment. What about your thrombosis model? What is that? Like, how do you test that?
Yeah, so we have a pump which pumps. You can even pump blood, or you can pump washed platelets, which is the platelets purified out of the blood, and you flow. So in the case I showed, it was washed platelets. You flow these platelets over a surface, and we've done it over a collagen surface or plaque, so actual plaque material taken from patients. And you can then image the actual platelets and the formation of a thrombi, and you can look at the size and you can look at different markers that are being expressed to see if those platelets are activated or not. So essentially, that's how it works. Yeah, it's it's a very useful kind of ex vivo model.
Okay. Okay, so I'm just told there's some questions coming in for Shubhangi. I don't see those questions, unfortunately. Let me have a quick look. Okay, I can't see the questions yet, but let me ask Shubhangi a question anyway. So is there a way to predict off-target sites in your analyses? So if you have a drug that's going to hit this ADP ribose binding site, is there a way to sort of mitigate against it having an effect on general cell metabolism, for instance?
Definitely. That's the one thing that Darren and I were discussing, like what if it does bind to the human ADP binding pocket and it could cause toxicity. So that's the one thing that we're going to look at. And we thought of kind of performing multiple sequence alignments of, the human sort of helicases or, the proteases, just to see if we do have those conserved residues in us as well. So that's the point of investigation that we're going to work on very soon.
Okay, very good. And then one more question would be, I mean, have you yet thought of a sort of how to work out the efficacy of these drugs on the virus itself? Is there some preclinical test you have that?
Not at the moment. Like, it's just the CASH organization kind of have the assay, information, and we have no clue as to what will be done after we do find hits. So I guess the preclinical stuff might be something that the CASH organization will be looking at. So we don't know.
Okay.
Yeah.
And for Shubhangi, there's another question here. I'll just read it out. It says, I might have missed it, but when you were identifying potential alternative binding sites, was there a criteria you used when certain areas were highlighted to define another potential allosteric or alternative binding site?
Okay, yeah. So as a part of the challenge, we were just allowed to kind of target the ADP binding pocket. So as I had mentioned, like these 6 pockets kind of covered half of, the ADP binding pocket. It was just the pocket 4 that was not fully covering the ADP pocket, but half of it. So we just kind of revolved around the ADP, like the central binding pocket, and nothing that was at the back or other allosteric pockets. So as a part of this challenge itself, it is just the central binding pocket that we were targeting. But as a part of my project, I'll definitely look at other allosteric binding pockets.
Okay.
Yeah.
And another question for you, Shubhangi. You mentioned that the compounds are targeting non-structured regions of the protein target. Have you tried to explore the binding pockets in different conformations of the target, or dock the hit compounds to different conformations of the target?
Yes, so it was for pocket 5 and pocket 6. So we had 2 different conformations of the protein, one with like a closed tunnel and other with an open tunnel. So it was quite necessary for us to dock like the compounds of these 2 different conformations, and we did that with pocket 5 and pocket 6.
Okay. I have a question here for Kathy. Would there be any particular tips to produce and purify membrane proteins or proteins that associate lipids?
So that's more of our team in Sweden who work with membrane proteins. So my only knowledge about working with them is like the standard things we're taught at university, so using adding a detergent or something. So I'm really sorry, but I'm not going to be able to provide much help for that.
Yeah, they're particularly difficult, isn't it, expressing membrane proteins? Like, it's very, very difficult. Okay, now a question for Alex here. Do you think the dimer interface identified previously is crystallographic? And with regards to the dimer interface you identified, what other methods could be implemented to confirm if this is the true biological dimer interface?
Yeah, this is a really good question I get asked all the time, and it's a hard one. So the first part, which is the previous dimer interface crystallographic, it could be. If you look at the original manuscript, they do really go and look into that, and they conclude that it's not crystallographic, it is real, but they didn't really follow that up. So it's possible that that one is a crystallographic artifact. It's also possible that our one is a crystallographic artifact, and more studies will be required. As for ways to do it, so I did show one experiment, which was fluorescent correlation spectroscopy, which actually directly looks at dimerization in the membrane. So potentially you could use that technique combined with some of the mutants that I made to see if that loop region is actually involved, is required for dimerization. That would be good. That would be a good kind of good evidence that there is a domain swap. The other way, which is another reason I wanted to move to the full-length protein, is this domain swap present in the full-length structure? And again, that would be, good evidence that it is real. But it's a very hard question, right? Relating the structures to function. Well, that's the whole point of this seminar, right?
Yeah. Okay. As we have no more questions, I'll just wrap up now. So it's a few things I need to say here. So thank you everyone for attending, and thank you to our speakers. You can continue conversation online, following at BioChemSoc and at PP Publishing on Twitter, apparently. Next up, we welcome suggestions for future topics and speakers to feature in the Biochemistry Focus webinar series. If you have an idea for a webinar in 2023, we invite you to submit a proposal for an upcoming webinar. If you can find more information about the webinars, propose your webinar, and watch previous recordings of webinars on the biochemistry website, which is www.biochemistry.org. So, you might have colleagues out there who'd like to do this, and and it'll be very welcome by the Biochemical Society. All of our upcoming webinars are listed on our website as well. And if you have missed any of the 50 or so webinars that we've already run as part of the series and would like to watch them again, please visit our website or our YouTube channel. The recordings from today's webinar will also be available to watch within the next couple of weeks. Join us on the 9th of May at 2 p.m. British Standard Time for our next webinar entitled Intellectual Property. Dr. Sarah Holland and Dave Vogt from Potter Clarkson. They will equip attendees with foundational information regarding intellectual property, with a focus on patents in the context of the research scientists and scientist founder. What it is, how to get it, and what to do with it once you have it, and be available to answer any questions that you have on patents and intellectual property. Finally, I want to highlight that it's more important than ever to stay connected and engage with your fellow molecular scientists. It's truly a fascinating time, particularly with the liaisons and collaborations between academia and industry at this time. Join the Biochemical Society's community of researchers and specialists and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. So you're very welcome to visit the Biochemical Society website and explore it. So last then, just again, it falls to me to thank everybody involved. Thank you for participating. Thank you for your questions. Thank you to our speakers, and thank you to the Biochemical Society. So bye-bye everybody, and thank you.
Bye, thank you.
Thank you, goodbye.
Thank you.